Protein synthesis is a two-step process. The first step is transcription, in which the sequence of one gene is replicated in an RNA molecule. The second step is translation, in which the RNA molecule serves as a code for the formation of an amino acid chain. An enzyme called DNA polyamorase reads the DNA and builds a strand of the RNA that serves as a mirrored sequence. For example, where there is a G in DNA, there will be a C in the RNA and vice versa. Once the whole gene is transcribed, the RNA molecule detaches. The RNA, called messenger RNA, then leaves the nucleus. Then, a ribosome finds it and they attach. The nucleotide sequence of a gene translates into a polypeptide, which is an amino acid chain in a 3-to-1 fashion.
Here's an example, like a drawing of DNA replication. At the top you can see is the leading strand, and the bottom you can see is the lagging strand. And in DNA replication, the thymines are replicated, or thymines, or however you say it. So yeah, this is just a quick example of what DNA replication actually is. Like I said, at the top you get the leading strand, at the bottom you get the lagging strand, and you can see that at the top, on the very top, that strand, it's 5-3, but then at the bottom it's 3-5. And then when you look at it like on the inside, the leading strand, it's 3-5 on the top, and on the bottom, for the lagging strand, it's 5-3. In DNA replication, there are 5 different components that make up what DNA replication is. Those being helicase, primase, primers, DNA polymerase, and ligase. There are different functions to each of these components of DNA replication. So for helicase, these are enzymes that bind and even remodel nucleic acid or nucleic acid protein complexes. For primase, it synthesizes short RNA sequences called primers, and they serve as the starting point for DNA synthesis. For primer, it provides a starting point for DNA synthesis. For DNA polymerase, it accurately and efficiently replicates the genome. For ligase, it is capable of catalyzing reactions of joining two large molecules by establishing a new chemical bond. Those are like the explanations of the functions of each of these components that make up DNA replication. For the DNA extraction video, that will be in the video, and it'll kind of explain everything in there. And then for DNA gel electrophoresis, DNA is negatively charged. Therefore, when an electric current is applied to the gel, DNA will migrate towards the positively charged electrode. Shorter strands of DNA move more quickly through the gel than longer strands, resulting in the fragments being arranged in order of size.
A polyamorous chain reaction is used to make many copies of a specific piece of DNA. Restriction endonucleases are bacterial enzymes that cleave DNA into fragments at specific recognition sites within molecules known as restriction sites. Phosphodiester bonds are the links between the nucleotides. The enzymes do this by coming into contact with the DNA sequence with a shape that matches part of the enzyme and wraps around it, breaking the strands of the molecule.